Today is 25th of March, Wednesday, and this is maybe recorded lecture 4. I can't remember the numbers, but we're right on schedule with the date and the time of the lectures. And each time I'll put the dates, so because many of you are watching these asynchronously out of schedule. But today, we're moving off of stomates and the movement of carbon dioxide to get plants to grow, and we're moving into respiration. So the first thing I put up here is stochiometry, and as by way of introduction, there's 2 sections in the textbook on crop respiration, chapter 5 and 6, 2 different chapters out of different books. Honestly, if you read sections on respiration in a plant physiology textbook, that would give you an excellent overview as well. But we're going to talk about whole plant physiology in here, which is not necessarily in plant physiology textbooks. Today's lecture is more on the basic principles. So I like to think about the stochiometry for starters and just keep drilling this. And if we do CO2 plus H2O, CH2O plus O2, our most important equation in the world. And now you get a 2 for 1, because if you put the arrow this way and put respiration, you've got the stochiometry of respiration. When it goes this way, of course, it's photosynthesis, and when it P and R and runs backwards. But let's extend this a little bit. This is the stochiometry. Let me, even before we say this, combustion and respiration have the same stochiometry. When we heat something, if you burn wood, you would burn carbohydrate in the wood, mostly lignin and cellulose, but it's still CH2O, and we would get CO2 and water vapor going up the chimney. But when it's combustion and we're going this way, we get a lot of heat. And that's the whole point of combustion, is to heat your house, and the heat produces mechanical energy, makes a car go down the road. That's fast combustion. This is running really fast. Of course, if it's heating your house, we'll put this in red, then it's CH4. This is natural gas. Methane, which is the primary component of natural gas, but the stochiometry is still the same. This is all combustion. If it's respiration, now let's write this. We'll put this in green. Same thing. CH2O still requires oxygen. Going this way, CO2, H2O. Now, respiration is going to give us heat. Same thing. But here's the difference. The whole goal of respiration is actually to minimize the heat and get, I should put this in red, ATP, or ATP equivalents, because the whole goal of this is to get high energy intermediates to synthesize all kinds of molecules in the plant. Respiration is not a loss of carbohydrate, it's a conversion of carbohydrate into other things. And in the next couple lectures, we'll talk about the efficiency of that and what it does. But for now, this is respiration. Colon. And so we're trying to get ATP. Combustion, you never get any ATP out of that. I guess we should write this just the way we did before. We have lots of colors today. Remember how we wrote photosynthesis is CO2 plus H2O going up here to CH2O plus O2, big, and all driven by photons. Boom, driving this up the hill. There's the diagram of photosynthesis. Now if we add to this, we could say, doop, this is going back down to the same CO2 plus H2O to the same end products, but now we should put ATP. So makes carbohydrates, turns the carbohydrates into energy compounds, ATP, that's respiration in a simplified version. And of course the goal is to get just as much ATP as possible out of this. We always get CO2 in water, but we'll put on here ATP and heat. And the heat is, for a plant, is almost completely worthless. Anything that goes to heat, it warms the plant, but it's minimal warming, where the goal is to get lots of ATP. So we're going to talk about what can we do to get ATP and minimize the amount of heat. So on this exact topic, 3 types of respiration. Let's put these 1, 2, 3. When Rubisco backfires, as it does when you get oxygen in there, we call that photorespiration. And I guess I could draw my little diagram here. Rubisco and oxygen gets in here and it backfires and it makes CO2, and that's inefficient, not useful, but it makes CO2. And the definition of respiration is a biological process that makes CO2. And photorespiration is called respiration because it makes CO2. But this happens in the chloroplast, and it's not helpful. 2 and 3 are what we call useful, that's a U, useful, and not useful. Now, what if this is not useful respiration, but these 2 both take place now in the mitochondria of a cell. Let's do a cell. Let me see. Here's a cell. It's always giant. Here's the nucleus in the middle. We're going to put a chloroplast in here. C for a chloroplast. And of course the light comes in here, photons. And this makes carbohydrate. And it goes down to, and this is, if this was a real class, I would be asking the students, another organelle. What's the name of the organelle that burns off all the CO2? Should have a t-shirt with an M on it. Mitochondria. The chloroplast fix the CO2, and the mitochondria burn the carbohydrate, and of course they give off ATP. So in the mitochondria, all this, both the useful and the non-useful is happening, and this is classic respiration. But there is a somewhat unusual biochemistry that manages to not make any ATP. It burns off carbohydrates and only makes heat. So there's a biochemical pathway that makes a lot of heat out of this, and we call that not useful. Respiration. It doesn't have any special name, but it's fascinating. You know, why would a thing make, well, if you were freezing, you could respire away to warm, but this isn't the reason. Here's an article, happens to be from the New York Times. There is a few species, and the most notable one is lotus. This flower comes up, it burns off carbohydrates like crazy, it warms itself, and that makes all these odors, and it attracts insects that pollinate the flower. So there's some reason for this respiration that only makes heat. And of course it's so interesting that not only does it attract insects, but it attracts a lot of scientists too, because we want to go in there and figure out what's going on. So this not useful it's interesting, but it's rare. Everything we're going to talk about now is just useful because it makes ATP. ATP. And of course it makes NADPH, high energy, charged up energy molecules. That's all the useful. So, by the way, I'm going to draw you the cycles. In preparing for this, I get out my human physiology book, and the respiration in people and in plants is exactly the same. The biochemistry is the same, so once you get it for one, you know it for the other. And guess what? This human physiology book is loaded with chapters on respiration, the biochemistry of respiration, the efficiency of respiration. But there's not one single chapter in this human physiology book on the Kelvin cycle and photosynthesis, so it's quite a simple book. But does it ever have a lot about respiration? Here's a page from that book on biochemistry of respiration. And if you were taking a plant biochemistry class, look at all these arrows and cycles of things. There's a ton of biochemical intermediates that are very well studied, but you're going to be pleased to know that we're not going to talk about the biochemical intermediates. We're just going to talk about the efficiency. So at this point, what I want to do, I'm going to erase this little diagram and substitute it. I'm even going to erase that. We get lots of room here. Let's see, so put it in green. So we spent a few weeks talking about photosynthesis, and that's a bunch of intermediate compounds that result in the fixation of CO2. We didn't talk about the compounds, but these series of compounds have the circle of this that adds one CO2 each time, a bunch of biochemistry, has a name, and it's named after the person that worked it out. That person plus all their graduate students. You remember the name of that? It's a cycle.
Kelvin cycle.
Kelvin. So, this is photosynthesis. Here's Melvin Kelvin. He was at UC Berkeley. He died about 20 years ago. Look at his students have white shirts and ties in this picture. They must have dressed up for this because they can't believe they wore white shirts and ties to the lab. But this is old instrumentation in this picture, but they're working out the biochemical intermediates in here. It's a wonderful story of how they did all this. But there's a second cycle over here. Let's put this one in blue. This is also called a cycle. This is photosynthesis. And this one is respiration. What's that cycle named after? Ah.
Krebs.
Excellent. Krebs. And here's a picture of Hans Krebs. I had to look him up on the internet. As a young scientist, 30 years old, and then here he is, 70 years old. He died in 1981. He was a Dutch scientist, but these are both famous cycles: photosynthesis, respiration. This, by the way, is also called the citric acid cycle, but I like naming them after people. So I guess there's no circle. That's the Einstein cycle. We have to think up something that we can attribute to him. He's already got plenty of laws and equations named after him. So in a couple of minutes, I'm going to work out all of plant biochemistry simplified with the efficiencies using these two circles. And we're going to start with a review. I think I got enough room in there. We'll leave it there. Here is sunlight coming in, where the whole thing starts. And this is photons. They're coming in to the Kelvin cycle. Now, I like to think of these as a wheel, the pinwheel turning. And if these photons hit this thing, I put this in green. See if we got a nice green color. If this is some kind of a pinwheel like this, the photons come in here, and photons don't have any weight, but imagine they do, and they turn the cycle. They turn the clock like this because they're pushing on the wheel. So over here is CO2, and these little cups that hold the photons are like double sticky tape. And they come along here, and the CO2 sticks to them on the back, and it gets picked up, and here we have CH2O. Now I'm going to erase the top. Give ourselves plenty of room up here to look at this. So that's a pretty simple view of photons in, CO2 getting stuck onto the wheel with double sticky tape getting picked up. We could do other things. Oxygen gets in the way. That would be photorespiration. But all these enzymes are cycling this around, and up in the top, high energy CO2. This is the starting input to the Krebs cycle. Photons are the starting input here. Over here, it's carbohydrates that are, and the Krebs cycle has the exact same thing. We have little cups on here. And if I'm using brown, the CO2 comes in here to these cups and pushes the wheel of the Krebs cycle around. In the case of Krebs, over here we have ADP, adenosine diphosphate, which is basically a discharged battery, and this sticks on here, same way that like the double sticky tape. And up here, we get ATP coming out the top. And the CO2 is going in. This is a really simple diagram, but it encompasses a concise summary of an enormous amount of biochemistry. But it is missing one key thing. Before this carbohydrate goes into the Krebs cycle, there is intermediate biochemistry. The CO2 goes in up here. That takes, this is hexose sugars like glucose, fructose, takes those sugars and starts to metabolize them before they go into the Krebs cycle. Anybody remember the name of that? Once I put it up here, everyone's going to go, oh yeah. It's on the handout. Glycolysis. Let's put that in red. Had you heard of that before? I know that you have. It's okay. Boom. Why is this important? This is a kind of an add-on to this whole thing. So now this curves around, goes into glycolysis. By the way, this takes hexose, 6CH2O. So we probably should say, do something like this. 6, 1, 2, 3, 4, 5, 6. 6, this has to turn 6 times to get 6H2O to turn this. So why is glycolysis such a big deal? Why do we have it separate? Remember how this needs oxygen to feed it? This needs oxygen to feed this. Without oxygen, this wheel can't turn. It's a critical input to this. What happens when you run really fast until your legs burn? Lactic acid builds up. And lactic acid builds up because there's not enough oxygen to have aerobic respiration. This cycle is aerobic respiration. It absolutely has to have oxygen to turn, but boy, is there a lot of places in the world where we need respiration and we don't have oxygen. And the best case with people is lactic acid buildup from running really fast. And you get so much lactic acid, you can't keep going. Glycolysis does not need oxygen. It can run in the absence of oxygen. Wow. So underwater, and all organisms have glycolysis. Underwater, we could get energy compounds without oxygen. It's like we could become a fish or something. It's just run glycolysis. Here's the problem. The Krebs cycle, when this runs from 1 mole of hexose, let me make sure I got my, 1 mole makes, am I going to have enough room? I think so. 30 ATPs. That's a lot of ATPs. The glycolysis, when it is forced to run without any oxygen, ATP, dun, 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 2. Whoa, that's pathetic. It burned up all this carbohydrate and we only got 2 ATPs out of it. So this is horribly inefficient if we have to run this without oxygen, but it still runs. And for lots of organisms, it keeps them alive because it doesn't need oxygen. Where's all the energy go from this? How do you think we make fermented beverages? The whole industry of limiting oxygen means we get glycolysis and a whole bunch of intermediate compounds coming out of it. This is how we get alcohol. We prevent the Krebs cycle from going and run glycolysis. Flavors and compounds of things. Man, what would cooking be if we didn't have that? We reduce oxygen to get all these interesting intermediate compounds. And I suppose I could draw those up here. Let's say in people, lactic acid. But alcohol. Just flavor compounds, all kinds of things that from food production that come off of limiting oxygen. So this is no oxygen without O2. If we have oxygen with O2, now even glycolysis gets more efficient, so we get 6 ATP. With oxygen, we get 6 ATPs. Without it, we only get 2. So the sum, after this carbohydrate goes all the way through and charges up ATP, 36 ATPs. That's the total ATPs that we can get out of respiration if oxygen is not limiting. We've already gone through how much CO2 is limiting for photosynthesis. It's big time limiting because, where's my jars here? Here's oxygen, here's CO2. So this is limiting all the time for photosynthesis. How can this be limiting over here? It's 600 times more oxygen than CO2. Here is the central problem. The movement, the oxygen has to get there by diffusion in the last steps, and the diffusion this is true of all gases. It's true of CO2, it's true of oxygen. In air, in water, diffusion is 10,000 times faster in air than water. And this is a little bit like if you were going to go to Smithfield, you could get there a lot faster in the air than in water. You'd have to swim to get there in water. But it's an amazing difference. So all this biochemistry happens in water, but we have air pathways to get it right close. Even in a leaf, we have air pathways. If we do a leaf like this, here's the stomates. A leaf is 50% air. A leaf seems like it's solid, but it's 50% air, so the CO2 can go everywhere in a leaf. Now, let's take one of Wyatt's potatoes and tell me how the oxygen's going to get in that potato because it's all water. And the answer is real slow because there's so much water to go through. How about a carrot? Something big and massive that needs oxygen. It's got to have air-filled pathways to get there. Why do we have a whole bunch of classes on how to till soil to fluff it up, because we need air-filled porosity in soil so the oxygen can get there. If the soil gets compact, the oxygen can't get to the roots. So the primary reason we till soils is to fluff them up so there's more air in them, so they're more like a leaf, so they can breathe. So getting oxygen's a big deal to this. Here's the analogy that I've used that helps this soak in. Let's say we were going to have a contest of how long it takes for a molecule of oxygen to get to the surface of this whiteboard. And I'm going to draw this. I'm going to keep these caps on. Let's see. Draw this right here. Here's the surface of this whiteboard, and we're going to start a molecule of oxygen, shown in red, 1 centimeter away from the board, but we're going to have a contest, and the other oxygen molecule gets to arrive at the board in air. To make it so they got here at the same time, how far away would we have to start the one in air? If this is 1 centimeter in water, well, 10,000 times farther, 10,000 centimeters, and that is equal to 100 meters. The one in the air would have to start somewhere over there in the ASTE building, so they got to the board at the same time, 100 meters away to get there. That's just amazing. So if we drew this to scale, this one would start over here, and they'd get to the board at the same time. That's the power of this oxygen by diffusion, and the payoff is 36 ATPs instead of 2. It's a huge payoff for getting oxygen there at the right place. Now, I'm checking the time. I'm going to show you a picture of how plants deal with that. This is a picture that is in all, I don't know about all. There. It's a picture that's in many plant science textbooks. I think Jennifer McAdam has a picture just like this in her plant structure book. This is a picture of a cross-section of a root. Let's look at this one first. This is the xylem and the phloem in here. Here's the cortex. It's a picture of all the cells. The whole root would be like that big. A healthy root. Classic picture. Look at this picture. This is the same thing. Here's the xylem right here. Look at this. This looks diseased, right? We should better call the plant pathologist. Something's wrong with this root. Erenchyma. That's some Greek words, means, I think it must mean biological snorkel, because these are air-filled tubes inside of a root that helps the roots get oxygen when the soil is flooded. Most plants die when the soil is flooded, but if they can make a biological snorkel called erenchyma like this, now oxygen gets there through the inside of the root. And if you can spell erenchyma, you're pretty much ready to graduate. A-E-R-C-H-Y, air. No, it's not even right. I don't write it's A-E-R-E-N. Air, ink, C-H-Y, Look it up. I think it's Greek for biological snorkel, but it means these air-filled tubes in the roots. And they're tremendous. And plants can grow normally in flooded soils when they can make them. Now, some plants make these real readily. Rice makes them all the time, and rice takes flooding. All plants need oxygen to their roots. They just get it in some other way. Corn makes these really well, but lots of other crops never develop the capability to make them, and you flood the soil and the plant dies. They just can't make a renkema. It's amazing, amazing thing. Here's a related thing. Some years ago, by the way, here, I'll put this in a handout and send it around, this picture. With oxygen, flooded without oxygen. There's a little figure caption here. Some years ago, we were studying rice, and we had an accident, and we were germinating it in Petri dishes, and a worker flooded the Petri dishes, like with a centimeter deep of water. And when that happens, this is on some filter paper, when that happens, it would kill the seeds. They can't get oxygen, they die. Rice, the controls on the filter paper, grew nice long roots, everything was fine. Look what happened when they got flooded. The rice was able to run glycolysis and get enough energy to send a little tiny shoot tip up to the top of the water, and as soon as that tip got to the top of the water, it could get oxygen. And then it germinated and grew fine. We wrote a manuscript about this. It's unique to rice. It's the only crop that can send up a snorkel to get air, at least to my knowledge, and grow. But that tells you something about how important oxygen is. This is 10 to the 4th power difference in diffusion rate and how fast the gases move. It's true of CO2, it's true of oxygen, it's true of all gases. Let's put some efficiency numbers on our chart. This is I'm going to send you a handout of this, but if we do Kelvin cycle, make sure my numbers, we got output over input for efficiency. Photosynthesis, PSN, 1 CH2O per 10 photons. Now, this is close to it's pretty much the theoretical minimum for this, and this is times 490 kilojoules per mole, and photons, if it's white light from the sun, is 550 nanometers, is 217 kilojoules per mole. So we multiply these together, and you get 490 over 2170, and that is 22% efficiency. Excellent, but that's also theoretical max. Now we do photosynthesis 1 to 20. Now this is with photorespiration. This is a value that would typically happen in the field, and everything else is the same, but this is 11% efficiency when the quantum yield is 1 to 20, or 0.05. So here's a range. Photosynthesis, as a rule of thumb, we can say it's between 10 and 20% efficient, depending on temperature and very much depending on availability of CO2 and nitrogen, if everything's right. All right, how good's respiration? We can calculate the same way we're doing it here, the energy in the carbohydrate, that's the denominator, and what we need to do for I'm doing that in red, energy out is ATP. And energy in is the energy in CH2O. And it turns out, calculating Gibbs free energy, we get about, where am I going to put this? Right here. 31 kilojoules per mole of ATP. I'm going to erase this. We got the diffusion part committed to memory now. There's the energy conversion, because we want to get this in energy units. And if we use, for CH2O, this is a hexose now, 480 kilojoules per mole of CH2O. There's our two multipliers to put ATPs into energy units and get the glycolysis in energy units. So the numerator over here, let's put this over here. With oxygen, plenty of oxygen, not limiting at all, everything runs perfectly. We get 36 times 31, let me make sure, yep, 31. So the people use very slightly different numbers for this, but 31 is a round number, over 6 times 480. 6 CH2O times 480 kilojoules per mole. And this comes out to be 1,116 over 2,880. Boom, 39%. So, respiration with oxygen's 39% efficient. This is in textbooks, this same approach to calculating efficiency, better than photosynthesis, like double the theoretical value for photosynthesis and almost 4 times the typical efficiency of photosynthesis. But look, all it had to do was break down carbohydrates. Photosynthesis is pushing it uphill, and respiration is just breaking it down. So it ought to be more efficient because it's able to start with high energy sugars. It doesn't have to start with photons. So that's great. 39 to 40% efficient for photosynthesis.
Why did we use 480 kilojoules for CH2O there?
And in the other ones we use 490. This is a value for mixed sugars. It's very slightly different. And you get, depending on the type of sugar, you get a slightly different number right here. There's fructose, hexose, so it's an average. Yep, that's a good question because I mean I should work this out and just make them the same number to keep the math simple. But the reason you get slightly different numbers is slightly different kinds of sugars. It's a good memory because we did 490. Yeah, there it is, right there, 490. How about no oxygen? Let's do the same math. Put this in black. 2, no oxygen. This would be anaerobic. BIC, anaerobic Greek for no oxygen. No O2. 2 times 31 over 2880. And this comes down to be 2% efficient without oxygen. So low efficiency. So when you draw this like this, we get efficiency of photosynthesis on one side and efficiency of respiration on the other side. This, so you can follow this, is a handout that's coming that takes what I wrote on the board and does it, and then does the math down here that we just put on the board. So it helps to show the efficiency of of both of these processes. I think this is a good stopping place for the introduction to respiration. Now, here's the next question, and we're going to pick this up on Friday in the next lecture. If this is 39% efficient, the maximum, when we have plenty of oxygen, remember, it was right here on the board, we put carbon use efficiency as a multiplier in our whole system on this. And I'm out of room. CUE, carbon use efficiency. Remember the number we put in there? It wasn't 39%, it was a bigger number. If you look it up, 60%. How, if this is a maximum of 39% theoretical efficiency, how do we end up in plants, whole plants, with 60% efficiency of capture of energy into other products? And the short answer is, this is 39% efficiency when it goes all the way to the bottom and everything turns out goes to ATP. But plants are like tinker toys. You don't have to disassemble everything to component parts. They do partial disassembly and they put the component parts back together. So off of all of this is spinning out all kinds of carbon fragments, and the plant reassembles them. And when it reassembles things, it can get a better efficiency than when it goes all the way to ATP. So this happens in our bodies and it happens in plants. We don't have to take everything right to the bottom and build it back up. We can take carbon skeletons, we call them, and reassemble them. And when plants do that, they can get better efficiency. That's where we'll pick this up next time. Good luck on the take-home exam.